by launching different joint transnational calls in the different research priority areas that are detailed in our strategic research and innovation agenda. Mainly, we covered different fields at research level, such as nutrition and healthy lifestyle, nanomedicine, cardiovascular diseases, and public health. And in the second phase of the partnership, we will devote it also to foster and support initiated by investigator clinical studies. I really hope that this webinar will be very successful and fruitful for all of you. And now I would like to provide the floor to my colleague Slomit from the CSO MOI partner of ERA4Health, that they are in charge of organizing this webinar. She will kindly introduce Professor Calum MacRae. Thank you very much, all of you.
Thank you, Christina. It is my privilege to open the first ERA4Health webinar alongside the esteemed Professor Calum MacRae. He is the Vice Chair for Scientific Innovation at the Department of Medicine at Brigham and Women's Hospital, an Associate Professor of Medicine at Harvard Medical School. He is also the leader of One Brave Idea, an initiative uniting experts from various fields to uncover the early stages of coronary heart disease. Professor MacRae will discuss building learning health system for discovery, translation, and care. And following his talk, we will have a time for Q&A session, so please feel free to submit your questions in the chat. We have a little bit of a technical problem, so I will share Professor MacRae's slides, so just bear with us for a second. That's not the presentation mode. Is it in presentation mode?
No, it's not.
It's in regular mode, but not fully, not in slideshow mode.
I think then it will be okay. Is it okay?
There we go. That's perfect. Thank you.
Right. Okay.
So good morning or afternoon, everyone. It's an honor and a privilege to speak to you this morning. And the title, as was noted by Shlomit, is Building Learning Health Systems for Discovery, Translation, and Care. Next slide, please. I'm going to try and give you a fairly high-level overview of our work across the spectrum from discovery right the way through to implementation. This is work that has been funded by both the multiple institutes at NIH, by One Brave Idea, which is a large grant from the American Heart Association, Very Life Sciences, AstraZeneca, and Quest. 4 companies have come out of this innovation program, but probably the most important conflict is academic self-interest, because it's the only one that you can't measure. And that's going to be a theme of what I'm talking about today, is actually the remarkable deficit of objective data in medicine and the need for that to really drive any closed-loop system that we might wish to build that can advance healthcare at the rate that almost every other field has advanced over the last decade or so. Next slide, please. This work, I'm going to highlight 4 people. Rahul Deo is a partner in this early work and is now in one of the companies. He's the Chief Technical Officer at Atman Health. 2 postdoctoral fellows who have just recently moved to faculty jobs, Wandi Zhu and Shinichi Goto. And then Max Hemilius, who's really pioneered a lot of the underlying information technology and analytics behind this. And I would highlight, obviously, that these are only a representation of a very large number of people who are involved and funders that have participated, listed on the left. And I need to include both Apple, Nestle, and the Leduc Foundation, as well as the funders that I mentioned already. So next slide, please. Let's get into the general discussion. So as we begin to think about how to build systems in biomedicine, information content is clearly rate limiting, whether it's in genetics, in risk prediction in traditional epidemiology or in drug discovery. And every entity, there are unidentified subsets of disease that confound almost all of these approaches. So, for example, in this middle ground here, there are lots of overlapping disorders between the very large effect rare alleles, the very small effect common alleles, which have not really proven very useful for drug discovery. Similarly, if you add an entire genome to traditional risk factors, you see only this last incremental delta in the predictive utility, the C index. And this diagram on the far right, looking at the discrimination between responders, those who have no effect whatsoever, non-responders, and those who have adverse response, is present on, I think, on every pharmaceutical company's website, but still now, almost 40 years after it was first postulated, has not made it to the clinic. And I think the fundamental problem with medicine is that data collection, especially in human phenotyping, is just too slow to change. There are cross-sectional collection of static or limited dynamic range data. Most of the information we collect is actually, unfortunately, largely subjective or semi-subjective. And most of the diseases that we think of as unique biological entities are actually aggregates of heterogeneous underlying disorders that, to be honest, were defined using medieval technology like the taste of your urine, which is the defining variable in diabetes. Importantly, and I think this audience is well tuned to this, almost none of the conditioning variables are ever measured. So we never really think about the time of day, the time of particular hormonal cycle, the extant nutrition or social determinants. We don't measure any of these. In fact, when we looked at this previously, one of the things that was stunning is that real estate transactions have more environmental objective data collected than most medical appointments. Next slide, please. So the rationale for digitization and for high volume, high velocity data is actually the ability to build truly closed loop analytic platforms. In almost every other setting in our lives, these platforms already exist and have existed for a long time. In one instance, for example, I showed a massively complicated brand new electronic health system to the CEO of a large retailer. And he smiled and said, that's roughly where our supply chain management was in 1986. And that's largely the case for almost everything in medicine. We wait for people to come to us. We don't have any longitudinal data sets. Most of the data are sparse. And there's almost nothing that we can actually do to improve that without reimagining what it is that we measure. The ideal situation, I think, is depicted on the right. This is just a very high-level overview of Uber's technology stack. And there they have high volume, high velocity, high diversity data inputs, structured algorithmic outputs in a closed loop system that's continuously learning. And then human in the lead, not human in the loop. And they're always A/B testing almost everything in their system. So there is a really dedicated architecture to establishing the ability to deliver the current state of the art, but in a way that is continuously learning. Next slide, please. The gap in medicine can be measured pretty straightforwardly. So, for example, everybody on this call differs by somewhere around 10 to the 6 or 10 to the 7 missense nucleotides, yet all of the phenotypes, including translational phenotypes that are rarely, if ever, measured in the clinic, add up to only about 10 to the 4 different metrics. So we're probably 5 log orders off in basic information content from the ability just to discriminate common variation at the genome level, far less the massive complexity at a transcriptional level, at a proteomic level, at a cell interaction level, or across timescales that range from microseconds to 100 years. And importantly, in all of this, I haven't even attempted to quantitate the exposures because I think they're just so enormously unbounded that it's difficult for us to imagine them. So this gives you a sense of just how far off we are in terms of our ability to manage and measure, or rather measure and manage the basic information content in medicine. Next slide, please. So can we move to a very different vision. One where we're actually using the genome. I actually quite like the genome as a organizing principle for human data. I think one of the biggest mistakes we've made in genetics is to think about it as a diagnostic tool. It really is a framework for understanding and organizing biological data. It's the only truly bounded data set in biology. What would be really nice is for us to be able to measure at a very granular level objective data, not just at the far right in the red part of the curve where people have already developed traditional medical complaints, but actually also in the preventative phase, which does exist in a couple of areas of medicine, including cardiovascular, I'm proud to say as a cardiologist, but ideally also looking back to even antenatal states, as well as through the earliest phases of wellness, so that we can begin to predict in a very discrete way trajectories for individuals and allow that to build pictures of evolving disease risk, to allow timed and triggered interventions, and importantly, to also enable real-time learning. Next slide, please. So how can we go about doing this? So we've spent a lot of time, we spent many years doing high throughput modeling, actually using the zebrafish as a tool for doing this. We essentially identified core inputs, genome anchored genetic inputs, made genetic models of human disease, did empiric screening in 96 well plates for both other genes and small molecules that would influence the output, and have used this to discover actually numerous drugs now that are in various stages of clinical development. But ultimately, the fundamental problem with a huge part of biology is the unknown unknowns, the lack of really broad information gathering at a scale and scope that might make sense. So can we reimagine the basic phenotypes that we're collecting? Next slide, please. So one brave idea was actually designed to do exactly this. You can just go through this slide because it has, I think, a couple of transitions, but it was based on the premise that the existing traits that we measure, blood pressure, hyperglycemia, dyslipidemia, and other diagnostic tests are actually not risk predictors. They're early disease phenotypes. And in fact, what we wanted to do was really to move earlier and earlier in disease to try and identify the first deviations from normal physiology. Next slide, please. And next slide. And so this allows us to redefine disorders in a very rigorous way. We had actually realized that this might be possible several years earlier when we began a fairly dense and broad deep phenotyping effort. We used everything from auditory evoked responses, stride length recorded by near-infrared cameras in the echo waiting room, down to mucosal cell biology to identify subsets of heart failure phenotypes. And we were able to show quite convincingly that many of these are actually abnormal at stages when traditional disease is not yet evident. And you might imagine in coronary disease, this would also be the case. We typically wait for the retina to become abnormal, wait for macrophage deposits to be visible to the naked eye. We look at drug response only in the context of having met the indications for intervention. And we haven't really built systematic approaches to trying to understand earlier and often latent phenotypes. Next slide, please. So just keep going. And actually, this is just a reference to the fact that there was, even in the mid-1970s, there was a series of manuscripts showing that vasomotor abnormalities are present in teenage relatives of those with primary coronary disease. But that type of perturbational phenotyping has never made it to the clinic. Next slide, please. So we began to think, what are the sort of unbiased computable phenotypes that we might collect? So obviously we want to have a focus on biology in humans or in human primary cells. We want to be unbiased as much as possible. We want to evoke new phenotypes. And ideally what we want to do is to do this under real world perturbations. One of the biggest mistakes I think medicine has ever made is that we don't record at a granular enough level the responses to drugs or to nutrition or to other environmental factors. If we can do this in a radically low-cost way, can we begin to make medicine and discovery part of the same industry? The other thing that probably has not escaped your notice is that once you start to add unifying metadata, such as a chemical stimulus or a physical stimulus or temporal coordinates, you begin to allow yourself to integrate the various layers in multi-omics, something that has really proven incredibly difficult in the last 15 years. Next slide, please. So we tried this systematically. We first of all looked at machine learning in the context of existing data, such as a complete blood count. We actually published in 2022 that if you have two serial blood counts more than 2 weeks apart, it's feasible for you to begin to outperform the predictive utility of many of the known specific disease tests. So you can start to look at the risk of acute coronary syndrome, heart failure prediction, PCR cabbage events, just by training. Oh, it says I've switched to a different language. It must have been very strong coffee. The most important thing about this is we're taking an existing data set and retraining it against existing outcomes. And by doing that in a iterative way, we're able to outperform the majority of specific diagnostic tests in clinical use in modern medicine. Next slide, please. This gave us, I think, quite a strong indication that we were going down the right track. I mentioned Apple. I'm the principal investigator of the Apple Heart and Movement Study. This obviously is a watch-based, an iPhone-based study with multiple independent sensors. Now over 200,000 individuals. We have access not only to serial transactions, biological transactions, but also to medical transactions through the Blue Button records, which a significant proportion of the participants have allowed us to get access to. And this allows us to begin to map early physiology from any of these sensors onto existing phenotypes. Next slide, please. The ideal would be to predict disease. We began to look at this in a systematic way, really to ask, is information content improved by simple perturbational interventions? You can see here for a whole host of disorders, you can actually have a significant impact just by adding mindfulness, the deep breathing routine, to the standard measurements. This dramatically improves the dynamic range, changes the predictive utility of the test, and suggested that if we were able to introduce unifying perturbations across almost any phenotype, whether it's a wearable phenotype, a physiologic phenotype, or a cell biological phenotype, we would add huge amounts of information content. Next slide, please. So we did the same next with echocardiography. We're able, instead of trying to identify the risks of a next clinical diagnosis, reduced EF or some other arbitrary intermediate step, we just took the current congestive heart failure stratification approaches, compared those head-to-head in a group of undifferentiated individuals with a baseline normal echo who were in a type 2 diabetes clinic. We were able then to predict heart failure admission with remarkable utility, just using machine learning, not trying to predict any of the intermediate steps, low EF, high EF, normal EF, mitral regurgitation, different forms of cardiomyopathy, but just going directly to the events. And what we identified was actually a single principal component that mapped with very high statistical significance to a single locus in the genome. And this genome actually is, this genome marker is in the middle of a ubiquitin ligase that is responsible for the trafficking of almost every sarcomeric protein in the heart. And we've shown now that this is actually a really robust target for multiple heart failure modifiers. Next slide, please. So we wanted to take a much more systematic and low-cost approach. In the last couple of slides, you have to have either an Apple Watch or you have to have had an echocardiogram. What's the most common test that is done in human disease screening and human medical management? So by a factor of almost 5, it's the complete blood count. And importantly, the complete blood count is typically performed using a semi-automated flow cytometer, very simple benchtop instrument. And we began to think, could we use this to screen for large effect size genes that were missing in the prior analyses, but were actually commonly important in clinical disease. So we looked in this middle ground, phenotypes and genotypes for which there was no known assay, or there was an important conditioning variable. And we could add that conditioning variable, whether it's an environmental factor, a drug, or a microbiome byproduct. We're able to test this, and by using ex vivo perturbation, dramatically improve the dynamic range of the underlying assay. Next slide, please. We went on and executed on exactly this. This is the baseline on the left, the baseline profile from a simple CBC cytometer. We added a panel of chemical and physical perturbations. You can see here on the right, this is an actual image with a completely new population or subpopulation of cells in the presence of a small molecule in a subset of individuals. And we can then bifurcate the population into very different subsets based on the profile of responses to literally hundreds of these small molecules. Next slide, please. What this actually, we found, allowed us to do was to create millions of low-cost digital phenotypes. We built this massively complicated matrix of phenotypes, and then all we did was really just train it against clinical outcomes in the electronic record. Mass General Brigham has something in the order of 25 million clinical records, and so we were able to take these very cheap, very commonly obtained tests over time and use these to show which phenotypes in the matrix that we had identified were not only associated with the risk of a particular outcome, but then when we looked at the primary genetics in an independent population, such as the UK Biobank, were also mechanistically involved in the underlying syndrome. And in fact, we were able, you can see this is just one set of outputs. We were able to build stratifiers that allow us to predict with almost 50% power those individuals who will develop gestational diabetes, as an example, even before they become pregnant. So we've begun to peel back some of the latent phenotypes that it's possible to create. And as I mentioned, I think we're only scratching the surface. Next slide, please. Importantly, this work, and I'm only going to show you one example, there were over 150 that we identified with only the first 30 perturbations. This was in Nature Genetics in December of last year. We were able to take undifferentiated risk, evoke perturbations. This, for example, was a simple time-dependent perturbation. We identified a novel pathway that leads to significant increase, about something in the range of 25% increase in the survival of neutrophils in the peripheral circulation. It's mediated by 3 common alleles, 2 in a glycolytic pathway and 1 in a fatty acid oxidation pathway. And the individuals who have the concordant alleles for all 3 pathways not only have this latent cellular phenotype, but are also very strongly predictive of a highly inflammatory subset of diabetes, chronic kidney disease, and peripheral arterial disease. And we've shown in vivo models and now with early drug studies that it's possible to interrupt this pathway very discreetly using predictive tools simply because of the effect sizes of these genes. In almost every instance that we've selected, the effect size is Mendelian or pseudo-Mendelian in magnitude. So it allows you to get at mechanism in real time and move very quickly to therapeutic discovery. Next slide. We can do this with almost any other tool. We did the same thing, and I'm only going to show you one quick example, and then I'm going to go to the opposite end of the spectrum. We were able to show very quickly that you could actually do the same with a simple benchtop microscope, added ion channel perturbations for blood cells. Next slide. And in this, we identified a clonal bias in hematopoiesis that is activated by hyperglycemia. You can see in the bottom panel here, there's a small, probably as much as 20% of diabetics have this outlier response. And this outlier response is the selection of piezo-1 overexpressing clones by hyperglycemia. And this phenotype actually is strongly associated with unprovoked deep vein thrombosis in diabetics and in non-diabetics. present in about 25% of all diabetics, and in a very high proportion, maybe as high as 90% of those who have a unprovoked DVT with no other genetic cause. So a really interesting and unanticipated finding based on an unbiased or semi-biased approach to phenotype discovery. Next slide, please. So you can imagine it's possible to do this at massive scale. We've shown already, just by using very simple, this was published many years ago, very simple training sets. You can do the same for drug discovery. And this is where most pharmaceutical companies are using their AI, is actually in matching chemical libraries to targets. But we believe that if you really want to build closed-loop artificial intelligence or even closed-loop analytics that do not require any deep learning, you want to make sure you're going all the way to the final endpoint, which is the clinical phenotype. So how can we begin to implement rigorous and robust high-volume data collection, as well as mitigate many of the barriers to systematic care at the patient-facing part of translational medicine? Next slide, please. Ideally, what we'd like to do, I forgot this slide, is to build a closed-loop effort. And this is just an example of what we did do with very limited closed-loop. We're actually able to outperform a 6 million marker polygenic risk score for coronary disease using only peripheral blood, machine learning, and the cost of a CBC is only $3.40 in the US. So really massive information content at very low cost. Next slide. What we're missing in clinical learning systems is actually patient transactions. Most of patient care is unfortunately highly variable. I've talked in the last few minutes about the discovery component of this. How can we build early phenotypes? But importantly, how can we actually collect data in the clinic that allows us to be able to make that leap from the clinic to the basic biology? Some of it obviously is the phenotyping I've just described, but we need to be able also to leverage the clinical transactions if we're ever going to connect this to existing care. Next slide, please. I was going to say, it's okay, don't worry. Variation in care is a huge problem. This is a paper from 2013, but the results were unfortunately identical in 2018, that fewer than 20% of all diabetics, type 2 diabetics in the US, were at goal for their hemoglobin A1C, their blood pressure, and their LDL. This remarkable variation in care is largely, I think, a function of the artisanal nature of medicine. Medicine is by design not systematic, but very local and dependent. It's been shown. I won't present the data because they're very well accepted and not mine. But the biggest single factor in changes in medical care is actually the retirement of existing providers, that it's a function of the generational transitions in medicine. Next slide, please. So it takes about 12 to 15 years for anything to be truly implemented. So we thought, could we do this in a way that would allow us to automate large parts of care and task shift away from physicians where the evidence base would allow it, to allow the right intervention at the right time with the right person in the right system? So we took physicians completely out of routine tasks. We deconvoluted traditional guideline-driven care into a series of fixed algorithms. We've subsequently gone on and parameterized these in a very rigorous way. And what we then did was to empower non-medically trained individuals, typically college graduates, to be the primary point of entry into the system. And so these individuals, with the assistance of a fully algorithmized care delivery platform, are able to rapidly implement guideline-directed therapy in a wide range of conditions. The scale factors from these pilots are of the order of 1,000 to 1 for a navigator, about 10,000 to 1 for a pharmacist, and depending on the condition, something in the range of 5,000 to 20,000 to 1 for a physician. They reduce cost by about 60%. We're not yet at the point where digital technology is rate limiting, and provider and patient satisfaction are actually very high, over 85% net promoter scores. What This work was all published. It was in Circulation in JAMA Cardiology 2 years ago. So I don't want to go into the details of the output, but just to go through the approach that we built with you, which is designed to allow iterative improvement and enhancement as we go through traditional clinical care without any external clinical trials. And so this also, as you can imagine, begins to allow you to do the sorts of real-world studies that have never been done, clinical trials in populations that were never accessible, A/B testing of the order in which traditional guideline therapies are implemented, which is usually historical, not based on efficacy or any biological rationale, and begins to imagine how you can really change the way in which care is delivered at a granular and very precise way, tailored to the individual, based essentially on comparisons between that individual and every other person in the system. Next slide, please. So the way that we did this was to begin to think about how we bypass different blocks. We had already done this in genetics. We pulled essentially all of the traditional diagnoses that would benefit from genetic testing. We built an algorithm that ran in the background in our electronic record, identified those individuals who needed genetic testing, and then just ordered the test so that the physician or the clinician only had to sign the order form, because we found that the first barrier was that nobody knew how to enter the requirements for the test. The next thing we did was we gave the information back not just to the physician, but to the physician and the family. We allowed the family to recruit remaining family members, and we used QR codes both at the physician end and at the patient end to drive not only the delivery of the results of the genome, but also the next phases of testing to begin to correlate genetics with phenotype. So essentially semi-automating and bypassing blocks using information technology in a systematic way. This was really the sort of antecedent of the next phase. Next slide, please, which is to completely parameterize the core components of modern medical transactions. So we took clinical transactions and we made them biological operators. We made them very uniform. We don't pre-specify exactly the order in which things are delivered because we've been able to parameterize them. That can change based on the data inputs. But importantly, the initial algorithm is modified iteratively by data. This allows us to deliver structured subroutines. It allows us to have transparent AI, so we don't need to go through the regulatory process at the FDA. It allows us to build collaborative care agreements that work in almost every jurisdiction that we've looked at. We're now, this effort has been commercialized. I'm not personally working in it, but the company that was started with this is now working in multiple different markets. And in this setting, you can essentially upskill individuals to the level of a specialist in multiple different medical specialties, with software encoding almost all of the evidence-based guidelines. Next slide, please. The other thing that I didn't mention was that this also allows comprehensive workflow redesign. So you can completely begin to address many of the burnout elements in modern medicine by distributing tasks not only to less trained individuals, but actually ultimately to the patients themselves. And as I mentioned, very high net promoter scores. Next slide, please. In fact, despite seeing the physician on average only a third of as frequently as they had previously seen their physician, patients in general felt more connected to their practice than they had done in the past. Importantly, the nice thing about this strategy is it allows you to devolve all of the effective and proven care to this platform while identifying key triggers that allow you to pop off back up to a physician or a more trained individual based on objective data or responses to intervening therapy. And this ultimately allows reinforcement learning of the entire network. Next slide, please. And I think we can just go through, I'm conscious of the time, we can just go through the slide very quickly, but this is just to say that by completely parameterizing everything, it's possible, including the diagnosis and the management, it's possible to build truly learning algorithms. And we have a couple of great examples of this, including implementation of a hypertension program in a largely housing insecure patient population that had never been included in clinical trials. We found that the majority of the traditional antihypertensives just did not work, but the system was able to learn within about 3 to 4 weeks that 6th and 7th line therapies were actually quite effective. And within a matter of 12 weeks, all 500 individuals were at goal. Next slide, please. This just shows that we get outcomes which are exactly what you would expect from the implementation of existing guidelines. You get substantial reductions in LDL, about 40% plus. You get almost everybody at goal within, actually within 6 weeks or 7 weeks. For hypertension, it's within about 12 weeks to 16 weeks for dyslipidemia. And we're now doing this across multiple medical conditions, including chronic heart failure, end-stage renal disease, chronic arthritis, and beginning to build out also activities in behavioral health. Next slide, please. So importantly, this type of systematization allows you to begin both data collection and care delivery in multiple different venues. And I do think you're beginning to see this in the US. It may not have reached Europe or elsewhere, but you're starting to see healthcare be delivered in not only in traditional clinical venues, but also at home, in supermarkets, in pharmacies, and even in cars and on television. So I do believe that there's a general trend to completely reimagine the workflow of care that's possible when the algorithms are deployed in a provider agnostic fashion. Next slide, please. We're almost there. Ultimately, the goal of this type of platform is to use human assays, whether cell-based assays or clinical information, to derive specific responder, non-responder subsets, discrete biological subsets for drug discovery, mechanism-based cohorts for either implementation or for clinical trials, and ultimately a continuous closed-loop optimization of not only basic discovery, but also translation and implementation. Next slide, please. So in summary, I've picked really just the two extremes, the early discovery and the late stage implementation. But the goal was to try to convince you that it's possible to systematize each of the steps. There are substantial increases in information content available at very low cost. Actionable disease subsets can be systematically defined, and this may even be how we redefine disease. Most of what we've done is really improve the way that we measure legacy medieval traits. We need to reimagine what it is that we think is a disease. I hope I've convinced you that automated care delivery is feasible. Obviously, there are significant risks and barriers. We've tried to do this in a way that eliminates unwanted and unwarranted variation to get you to maximal precision and to allow you to reimagine workflow around the disease and the individual rather than around the care delivery mechanism. And then finally, I think we've begun a process which will ultimately lead us to optimize all of the steps from discovery right the way through to late stage implementation. And if we can do this, I genuinely believe that we can reimagine all of the aspects of health and wellness. So I'll stop there and take questions. Thank you for your attention.
Thank you very much for an excellent talk. Before I read any question, I would like to ask, it raised question about regulation and data privacy and who actually owns the data. So I was wondering if you thought about maybe creating a kind of NFT, a non-fungible token, so the patients actually own his own data and can even use it.
Absolutely. So actually, I didn't dwell on this because I thought it was probably too much in the weeds, but we've built a completely blockchain-enabled platform that allows you to actually do conflict-free collaboration in the clinic. So I think one of the most amazing things from my standpoint is that I think medicine is the only area of human endeavor where the research and development and the actual implementation are separate industries. And so what we've done is by building information tools that allow data to be captured and delivered in a private and secure fashion, you can actually overcome many of the barriers to academic industrial collaboration. And in fact, we're planning to roll this out sometime in the next quarter at Mass General Brigham. We do believe that that's ultimately what you need to do. And I think your point is very well taken. That's exactly what every other industry has done. I mean, banking introduced these types of data workflows almost a decade ago, and medicine is just, as usual, 20 to 50 years behind every other field.
So why do you think the medical world lagging behind? Why there was no proactive actions about it?
Well, I think it's largely because it's a business model for medicine. Medicine is it's a one-to-one interaction between a patient and a physician. And we've sort of maintained this as a sacrosanct right for decades, actually for centuries. We're probably one of the only legacy medieval cults to survive. And you saw this during COVID. You know, we had literally 1.5 billion individuals died from COVID, were exposed to COVID. About 6 or 7 million people died. And the only useful information came from 2 cohorts, one in Israel and one in the UK. Almost everything else all the other information was thrown away because it's all in individual silos of either the smallest academic medical center, even in the US, the largest academic medical practice has only about 2.5 million individuals in it. So there are no really fully integrated data collection architectures. You know, most large data-driven companies are managing hundreds of millions of transactions a day. We're literally like corner stores. We're basically the last surviving hyperlocal industry. And when COVID came along, it was embarrassing. I was the chief of cardiology at Brigham and Women's. I tried for almost 3 years to change from 1% to 2% the proportion of consults that were virtual. And when COVID arrived and it suited the physicians, we were able to go from 1% to 97% in 1 week because it was in the physician's interest. So most of this is physician self-interest. And I've largely committed to trying to overcome that now that I'm old enough not to really care.
Interesting. So a question from the audience, a brilliant lecture. Can you tell how nutrition is assimilated to the algorithm?
So actually, nutrition is a really interesting area of quantitation. We spent a lot of time with Google, with DeepMind that Google had purchased. One of the original elements was actually efforts to try and use imaging to quantitate nutrition. What we basically decided, and we've done similar things with other partners, is that it's incredibly difficult to quantitate nutritional input. What we decided made more sense was to quantitate some of the small molecules within the nutrition. And so what we're able to do is actually to use nutrients. And I mentioned Nestle and Purina are a sponsor of some of our work. We've begun to use individual nutrients as perturbations in our latent phenotyping. And you can use either microbiome products or you can use magnesium. We've identified as a proof of concept, there's a very poor correlation between serum magnesium and the effects of magnesium in a clinical setting. We basically built a magnesium response assay, and we've showed that that's actually highly predictive of the response of arrhythmias or gastrointestinal symptoms to magnesium, and really does give you an index of not only your total body magnesium state, but the responsiveness. So that's the type of thing that I think will be feasible. But I do ultimately imagine that, and I didn't say it as explicitly as I usually do, but most of the data that we're going to collect in medicine is not going to be data, or are not going to be data that we think are important in the clinic. It's going to be other elements from the rest of your life. We can't afford to build the data collection architecture separately for medicine. I think we're going to have to use credit card transactions. I mean, to be honest, the the nutrition that you buy is probably the best index of the nutrition that you're exposed to. So that might be one indirect way of capturing it. But the reality is that that applies to almost every parameter in medicine. We're completely underdetermined compared with even the average bookseller on the internet in terms of our ability to understand our customers and our partners.
Thank you. So next question is, what you describe is the implementation of 2015 on-sheet vision on learning health systems, yet almost 10 years after, there is not much in place. What are the main challenges to scale up what you have described?
So we built our first hypertension, lipid, and heart failure program in 2012 when I took over as chief of cardiology at the Brigham, and mainly to help us to scale the practice. We were getting, with the advent of PCSK9, we suddenly went from a very small number of specialty lipid referrals to really huge numbers every week. But the main barriers to scaling inside the Mass General Brigham system were actually the competing interests of the existing business model. So it's very difficult for you to move care from the existing providers to a third party when you're currently paid only on the basis of the existing providers. And so what we've done, as I mentioned, is create a startup, Silicon Valley funded startup. Khosla Ventures were kind enough to support us. And we have begun now to implement in very specific financial settings, usually value-based care, very complicated care where it's really difficult to keep track of all the interactions, and in single-payer systems such as the UK. So we've completely built out the entire platform, redesigned it, as I mentioned, in a fully parameterized fashion, and are implementing worldwide. But the main barrier, to be completely explicit, is physicians. Nobody likes their gig to be disrupted.
Yeah, we are familiar with that. Any plans to implement that in third world countries?
Actually,
or have remote medical treatment?
We are looking at how to, we think we need to get to a particular scale factor. But we have been in discussions with a couple of sub-Saharan countries about the possibility of implementing in Africa, and also talking to partners in India about implementing this at scale.
Thank you. So next question is, can you address harmonization of data for discovery and innovation, meaning OMOP or the FEAR platform?
Yeah, I think, again, this is partly, I think we do need really robust data standards. I think one of the biggest challenges in medicine is we like them to be our own. And the reality is data that are truly useful are actually calibrated at the point of origin and are fully digitized at that point, and you don't need secondary data standards or curation. This is one of the things that has been remarkably effective, for example, in online transactions of any sort. But I do think we're going to end up using data standards that are not medical specific. I think they're going to be commercial data standards. I think they're going to be based on individual phenotypic points or individual metrics that we're measuring. We're not going to be able to enforce our own curation on existing data, which is what we usually like to do because it makes us feel like we're involved. The reality is it's an embarrassment that we actually have to standardize our outputs when we don't make any effort at all to standardize our inputs. I mean, the saddest thing about electronic records is they're almost no utility unless you have documentation standards. And I'm not aware of any large medical entity that has successfully implemented documentation standards using current physician workforce. So I think we have to get over ourselves. We have to start to think about where the data are coming from, work with the sensor or the input source to make sure that we're accepting standards that are useful for us, but also are useful for other applications of these data, because we will not be able to claim them as purely medical going forward. And in fact, I think electronic records will suffer the same fate. I do not think there'll be on-premises electronic records within a decade. Most individuals will be managing all of their own data using exactly the type of tokenized platform that you described, Shlomo. So your financial data is probably one of your best predictors of your medical outcomes, and yet we don't even begin to measure it.
Thank
The
you.
average shoe seller has better data standards than modern medicine.
Okay, I'm going to combine the next two questions. Do you think this project is another step for precision medicine? And can this system be used for reorganizing of health services, making that accessible to both the clients and health services providers in a large geographic area to interact between clients' specific needs and possible services for them?
I would love to believe that that's possible. I'm not certain how quickly it'll come to pass. It definitely is more precise. I mean, we've shown systematically that just by measuring drug responses in a systematic way, you can identify more precisely population effects that were missed in large randomized controlled trials. So I do believe that this will be more precise than the current system. On the other hand, when I was in medical school almost 35 years ago, we thought precision medicine was, 6 months around the corner, and it's still not really being implemented. So I'm not agnostic to the challenges in getting more precise intervention, but standardization, as Dr. Skorecki outlined in his question, is important. I just don't think it has to be medical standardization. We have to be in a position where there are rigorous standards, and then things become much more precise. We don't measure anything precisely enough to be of real utility. I think probably the best example of this that I'm aware of is life insurance companies identified the risk of tobacco in 1911. It took the medical profession until 1959 to document the same association. And importantly, the insurance companies priced it perfectly in 1911. The weighting factor for tobacco has not changed since 1911. We imagine that the data we collect are somehow have magical properties, when in fact just counting things properly and robustly in a uniform way has much, much greater utility.
So if you mentioned insurance company, aren't we going to see Gattaca, like the movie Gattaca, where babies are going to be scored based on their gene expression? Or
so I run a fairly large genetics clinic. That's my specific clinical area of interest, is inherited heart disease. My original work was in the genetics of hypertrophic cardiomyopathy and dilated cardiomyopathy. And the scariest thing is that, first of all, genetic discrimination is prohibited in the US, but the reality is the life insurance companies have a better estimate than any genetic test does of what your liability is for familial causes of sudden death. One of the most amazing things that's happened is that the introduction of the electronic health record has more or less eliminated family history and pedigrees from medical records. And because it's so difficult to record these using the technology. And so I hear what you're saying, but I actually think having predictive tools that you can manage your life on is much more important than worrying about whether somebody knows what is going to happen to you. I do think, however, we have to put in place really rigorous protections in terms of financial disadvantage, other societal disadvantages for individuals who have either genetic or non-genetic risk. I mean, many of us have huge environmental risk, and we need to think about how we factor that in. But ultimately, that's a societal approach to shared risk and shared liability, and obviously an important piece of what I think you're seeing being prosecuted worldwide in politics at the moment, where there's a dramatic tension between shared responsibility and individual responsibility that we have to re-prosecute every 70 to 100 years. And that itself is quite embarrassing that we go back to the same places every time. But I think we're hopefully going to be able eventually, with better information flow, work out how to balance that shared and individual responsibility more clearly across society.
Yes, I agree with you. And last question, how did you overcome many individual reluctances to be genetically tested? The fear of discrimination, and how did you manage to inform consent the process?
So that's a very interesting question. My experience has been that most people want to be genetically tested. Within every family, there is typically there are a few individuals who don't. I think some of this is whether there's actionability. So I think when I was a trainee, you used to have to consent somebody to collect their blood for HIV. You used to have to consent them to give the result back to them. And then there was a third consent form to put the result of the test in the record. And now people wear their HIV status on a band on their wrist. And the thing that has changed is that there's actually an intervention. And I think if we actually are changing the cycle time to intervention, suddenly a lot of the barriers to testing disappear. But there will always still be some. And so we make sure that we include the possibility to opt out, but we also blind the whole family. We built, you may not have seen it in the diagram, but we built a way of displaying relatedness without necessarily displaying who was who in the family. So you can't tell gender or the actual SIBSHIP in which individuals are in the representation of the pedigree that we built, so that any individual can either give their genetic test, but genetic data, but not tell anybody that they participated, or vice versa, not participate, but also not tell anybody that they did not wish to participate. But in the main, usually the good of the next generation is sufficient to overcome most barriers to genetic testing. And we did not find it difficult. I think there were only, out of the first 100 individuals, there was only one who refused to have genetic testing.
Christine, thank you very much for your excellent talk and for the early hour that you attend. I'm inviting everybody to our next webinar on November 11th about pragmatic comparative effectiveness randomized trials. You can find the link to registration in the chat. And have everybody Shana Tova, which is the Jewish New Year, and a good week for everybody.
I would like to thank you, Professor MacRae. Thank you very much for this great lecture and all the attendees. Thank you.
You're very welcome. It was great. Thank you for all your kind questions and comments. Thank you.
Thank
Good
you. And bye-bye.
Bye.
Bye.
Thank you.
Thank you.